is a special episode of the Stem Cell Podcast, ISSCR 2024 on day 3. Hey everybody, we are Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. We're back with our third episode covering highlights from the ISSCR 2024 annual meeting, which is in Hamburg, Germany. Arun and I have had so much fun attending and getting to meet all of you, but it's not over yet. If you're looking to find out what we're up to at the meeting, follow us on X at Stem Cell Podcast. You can also find episodes covering the 2 previous days of the meeting at www.stemcellpodcast.com or wherever you find your podcasts. Today, we'll be recapping day 3 of the ISSCR 2024 annual meeting. We're going to kick things off in just a minute, but before we get there,
as stem cell researchers, we are united by many things: a love of science, insatiable curiosity, and a desire to produce high quality research that will move the field forward. To learn about how stem cell technologies can support your quality journey at ISSCR and beyond, visit www.stemcell.com/ISSCR.
yes, we are united by many things, particularly here in Germany. We've had the best weather. I think the camaraderie and collegial spirit amongst all attendees is high. And everybody's enjoying themselves to a tremendous degree. And I think engaging with the science on a very high level. So I'm pleased with the way this meeting's gone so far. Already a huge success. And we're only at the end of day 3. Getting back to that, I started the day with the concurrence, and given the fireworks of 2023 meeting with all the embryo models, I had to get into the non-integrated stem cell models of human development there, underlining the non-integrated. I guess this is kind of deconstructing the human embryo model into its pieces and our constituents. I don't want to. It sounds creepy cutting human embryos up into pieces, but the talking about the cellular constituents, really, I would say, in this session. And I led off with a talk from Yasuhiro Takashima, who's from Kyoto University, who was using naive hematopoietic stem cells to model in vitro differentiation of extraembryonic lineages. And he did this in these bilaminoids, which are constructs made up of hypoblasts and epiblast. And he showed some mechanistic data involving IL-6 and JAK-STAT signaling and formation of the amnion, which is driven by the trophoblast. It was a nice, for me, illustration on how genetic engineering and human pluripotent stem cells are converging on this deconstruction of human pre-implantation development, even outside of the context of these human embryo models, like really getting down to those cellular constituents and the mechanisms that govern their differentiation and emergence. Then next for me, it was a Merit Award winner from Amanda Clark's lab. A grad student there is Ariana Arapor. And this was a really cool talk for me too. They kind of two-dimensionalize. I know 2D is like a dirty acronym. I don't know what initialism. It's no one likes to talk about 2D anymore since organoids came into the mix. But this was kind of like a 2D hybrid where they have this overlay of Geltrex. And a lot of really interesting tools, including this live reporters to indicate, with the SMAD1 fusion construct to show the activity of BMP, showing it had a role. And the takeaway from here, this is why I love this meeting, and I love the way they've embraced the trainees and showcase them, because it's not one of these talks you get at the plenaries where they're talking about like 10 years of work, even though it's a story from this year, or even unpublished data. It's really, the amalgam of so many years of work with these grad students. You see them getting these projects off the ground, and you see how it really takes these basic descriptive analyses. You have to observe the thing in order to form hypotheses that you can test. And I think a lot of graduate students are doing that. It's the eyes and the scopes that are generating the data that then we can use to create these frameworks that are testable. So I really like that. Next, I caught a talk from Madeline Lindberg Agerholm. My apologies to any mispronunciations. She gave a great talk, also a travel and merit award winner, a PhD student at Novo Nordisk, working with Josh Brickman. And this was a deep study. I mean, real deep mechanism which was showing the capacity for primitive endoderm to generate both embryonic and extra-embryonic lineages with all these methods. It was so cool. You ablate all the lineages, and the primitive endoderm alone was able to recover and create full blastocysts. Um, and it showed the mechanism as JAK-STAT signaling, and that the pluripotency, even at the epigenetic level, that pluripotency genes remain primed even in the primitive endoderm cells, so showing this kind of reversibility. A great story just published in Cell a couple 3 weeks ago on June 24th. So a really great showcase of students' data culminating in a high-level paper. Um, and then for me, it was on to another Merit Award winner, Young-Soo Kim, who's from Michigan, who is, we talked about the bilaminoid. This is a model of trilaminar embryonic disc. And another, I wouldn't say descriptive entirely, but a lot of descriptive data there. But here focusing on early hematoendothelial specification, which of course everybody knows is my favorite subject. And that brings me to the final concurrent that I caught, which was another beautiful story. I mean, I love the pictures and videos that were shared by Mo Ibrahim Khani from University of Pittsburgh, who was modeling this post-implantation development of hematoendothelial cells in the yolk sac. But the real key here was showing the organization of like these early embryonic type structures, right? Embryo models that then gave rise to these hematoendothelial cells. And I think this is the recipe. I mean, we've talked about before in terms of theory, but I think that this is the recipe that people are going to have to get to moving forward to get these definitive hematopoietic stem cells from pluripotent stem cells. And of course, that's going to take walking these cells way further down the path. But primitive hematopoiesis is the first milestone along the way. And I think the idea that we're able to reconstruct these embryo models is ultimately going to allow us to generate the complex niche that can support differentiation of a true hematopoietic stem cell. So I loved this whole session. I was dancing around a little bit, but the bottom line was the embryo models and application of those embryo models to understand cellular differentiation across many different lineages. So it was the perfect session for me to just finally, I've been moving between sessions, getting my steps in, but this was one where I could just settle down, settle in, and really be stimulated. Arun, what do you got?
Yeah, it's definitely the hottest area of stem cell research, as we've talked about a million times on the show. I'm glad you're able to go to that session. And speaking of models, that's what the topic of plenary number 5 was all about: organoid models of disease. First talk was none other than Clive Svendsen from Cedars-Sinai, my guy, my boss, I guess. He got kind of roped into doing a last-minute plenary talk last night at the reception there. That's what I was hearing. But he put together a last-minute talk that was awesome. In typical Clive fashion, covered a lot of things, but got the audience really engaged. Talking about all things modeling human disease using IPS technology with a focus on ALS, which is his scientific focus for many years. Talked about his journey of using IPS models, where he first saw that iPSC-derived motor neurons died in the dish, just like in the ALS patient. This was actually a really high-profile paper, I believe, in Nature in the late 2000s. You fast forward a little bit to Answer ALS, which is a very high profile and high capacity large IPS study, 1,000 IPS lines derived from patients with ALS. And the ultimate goal is to combine this massive data set with machine learning to tease it all apart. And shout out to the Cedars-Sinai Biomanufacturing Center, which has access to all these lines. So if you're interested in studying ALS and working on patient-specific IPS lines, reach out to them in the Cedars-Sinai Biomanufacturing Center. He's also talking a little about his organ on chip system, which I'm very familiar with. I work on heart on chip. He works on brain on chip, taking this technology from the Weiss Institute and applying it to develop this amazing blood-brain barrier chip that they published on a few years ago and have had a few follow-up papers on, and then touching a little bit on our space work that we've been doing over the last few years. And the timing is good. We've got a upcoming live seminar, live recording of the Stem Cell podcast with Kat Jameson and Davide Marotta, also fellow space biologists. So stay tuned to hear more about that. Moving on to Sharon Gerecht from Duke University, go Blue Devils, talking about the microenvironment regulation of vascular fate assembly and function, something that you're pretty familiar with. But this is more of an engineering approach to it, taking engineered hydrogels to model the microenvironment better, also looking at the effect of hypoxia on regulating endothelial cell differentiation, as well as other applications for hypoxia and development in cancer. Of course, we know hypoxia has a really important role in iPS reprogramming, but this is looking at other applications for that. Also developing these really cool hypoxic hydrogels that were layered in a precise configuration for optimal control of carbon dioxide concentrations, and also using enhanced angiogenesis in response to hypoxia. Other big picture of that talk was that extracellular matrix, as you might expect, impacts vascular development and regeneration. And overall, just a really neat talk about the role of the microenvironment to regulate different aspects of endothelial cell function. Third talk was extracellular morphogens and genomic variation, instruct cell type specification in human pluripotent stem cells. Flora Vaccarino from Yale University was giving this talk. Really great overview of using iPS derived organoids to model normal development. I mean, we talked with Clive and with Sharon, about the diseases, but this is looking more at normal human development and the brain organoids in particular to model brain development. The question here was how to better approximate human development using brain organoids and the differentiation of these cortical organoids. One thing that I really admired here was their approach on building better organoid systems with morphogen gradients. And just by patterning these brain regions along orthogonal sheets of Wnt and Sonic Hedgehog, they created this awesome system to examine the impact of these morphogen concentrations on how it ultimately gives rise to different composition of cells within the brain organoids. Really neat nuts and bolts paper there and study there. And finally, looking at the individual iPSC lines and how they differ in response to identical morphogen gradients. So really teasing apart brain organoid, cortical organoid differentiation at a level that we haven't really seen before. Last talk of that session for the plenary number 5 was Toshihiro Sato from Keio University, understanding the role of understanding human organ function and disease using organoids. Of course, this is a session on organoids. Focus here was on intestinal epithelium and how organoids can recapitulate the morphogenesis of intestinal epithelium, both in vitro and also transplantation of these organoids in vivo. Really neat talk on how to accurately replicate the pathology of tumors using organoids and having the same morphology in the real cancer tissue as you're finding in the organoid across multiple cancer types of the gut and the intestine, the stomach, and some nice unpublished data as well. We all love unpublished data on some hepatocyte applications, really great human adult hepatocyte organoids and how they have great function in terms of bile acid production and, of course, albumin production. I also caught an awesome lunch talk on the career paths and stem cells in low Earth orbit for obvious reasons. I was interested in that session. But also, it was really cool to see just how rapidly this field of space biology and stem cell biology in space has expanded in the last 10 years ago. I mean, I got started in this field as a grad student in 2003, and people looked at me funny. They thought I was crazy for sending these stem cell derived cardiomyocytes to the space station. And now we have many, many grad students from the United States and around the world who are working in this exact topic, looking at organoid production, looking at cancer morphogenesis and cancer phenotypes in space and how microgravity might alter those phenotypes. This is a great panel chaired by Davide Marotta and also Pinar Meski from Axiom Space, and talking to some of the students about their journey in doing stem cell research in space. And of course, we're going to have a great show on this exact topic coming up soon, but just warmed my heart to see the growth of this field.
I have to say, Arun, you're way ahead of your time. You had the foresight to see how big this would be, I guess, or maybe we're just interested, being an Alabama boy trying to get up there. But the bottom line is that what's driven the most interest, I think, at this meeting. Maybe a little aggressive to say that, but there have been outsized interest. I've been really surprised by how many of the young scientists in particular, I think, seem to have come to this meeting in large part because there's all these sessions that are really dedicated to this burgeoning field. So I'd have to agree with you, and it warms my heart as well to see how many people could give two S's about anything I have to say. They're just trying to get up there and talk about his space work. So I love it. On to the next. I mean, we had some concurrence after that. I was sitting on my butt for the whole morning concurrent. This time I was racing all around because I had very specific interest across many of these lecture halls. First of all, I was joking in the early days about how going back to basic here, where's all the clinical talks? There was a session here with the PSC-based cell therapies that, of course, I had to catch. First talk in that session was this autologous 3D engineered tissues. This was from Tamar Harel Adar, who works with Matricel, which is a 5-year-old company. And I mean, it's interesting when you get the industry talks, because it is kind of a different animal given the IP element there and the competitive element. In this case, the IP is related to 3D bioprinting and the application being spinal cord injury, which has been a mainstay target since day one. And I mean, we made a lot of progress, I think, but there's a problem that's not near to solved. The approach here is using these decellularized matrix that you generate from human omentum. So you take some omentum, you decellularize it, break it down to the cellular or to the extracellular matrix components, and then you can create an autologous hydrogel, which is a great idea. And then the idea that Tamara presented was seeding it with iPS-derived neural cells. Again, getting back to the different animal here, it's I think that a lot of the industry players are still kind of telling the story to the audience, like trying to sell the story, and maybe trying to hold back on the mechanism and the data because for whatever reason. But I would say, as exciting as the application and the endpoint here is, I think that this reminded me a little bit of early days in terms of like, showing that the cells have neural characteristics, but not really going into great depth in terms of like the molecular biology of the cells. But I will say, showed functional recovery in a mouse model, which is one of the major endpoint goals that we've always been looking toward. And it's worth saying also that Stefan, who is sitting in the audience, stood up. I think it took a lot of finesse to do this, because you don't want to get up in the meeting and be negative at all. But you do also want to try and you want to ask the questions. And Stefan asked, after everyone had their question, the last question is, he said, "What about the mechanism?" And I think that's what a lot of people were thinking. And I think we've reached a point where the field has matured, where there's a much higher bar. You can't just tell a story anymore. You actually have to have something that's not just going to be, safe superficially, but also efficacious. And I think what people are thinking more and more about is whether or not these things are going to be practical in humans. But still, a great introduction. I loved it in terms of like, how are we going to use these cells in people? And it was followed by a really, I think, excellent talk from a travel award winner, Eduardo Sozzi, who's from Malin Palmer's lab. And no coincidence that the science here was exceptional, and also really innovative in terms of the approach. This idea here was co-engraftment of dopaminergic neurons with other types of neural cells, supporting cells. And this is kind of affiliated, I don't know to what degree, I don't want to misspeak here, but it follows from the same, I guess, origin as the STEM-PD clinical trial that Malin and others are conducting, which is really exciting as a companion to the Blue Rock trials. So I think we're really moving forward there. And the key here was that when you took all these different types of progenitors, ventral midbrain, glial, ventral forebrain, striatal, there were two insights. One, which is that combinations of cells, and the output here was like looking at a lot of metrics for the cells, but also motor behavior recovery in this context of these amphetamine-induced rotation model. So actual real like functional improvement in these mice. And the insight here being that when you combine the midbrain with the forebrain, you get the best result here. You get enhanced maturation, and also the other thing in terms of like the target area when you have the interaction with the striatal cells, which is a target of these dopaminergic neurons, you get a diversity in the subtype identity. So I think it's a conceptual advance in terms of like how we got to think about these things that we got to optimize, and some of that optimization is going to be providing some of the other constituents of the niche and of the organ, and very well illustrated in this case in this excellent talk. I was running back and forth. I missed a couple. I missed the beginning of one. I missed the end of the other. I did catch the end of Meiyan Wang, who's a postdoc in Rusty Gage's lab, and I never get tired of seeing this like, big time engraftment of these glia, in this case, glia enriched cortical organoids. But you never get tired of seeing these mouse brains that are like all over human nuclei or GFAP or whatever it is. But it's just amazing to me that you can have these mice walking around with this tremendous innervation. It makes me really believe that these therapies and the capacity of these engrafted neurons from human pluripotent stem cells, the capacity for them to migrate and spread throughout and function, in this case showing increased maturation when they were injected in these glia-enriched cortical organoids, which we always talk about. That's a major challenge, is the maturation state of these. I think we're starting to crack it, although still clearly a long way to go, but a lot of nice data, and love the pictures. And finally, of course, I had to catch Katsuhiko Hayashi, who's like kind of my idol, young scientist who really broke open, along with many before him. Okay, of course, Azim Sarani and Mitsunori Saito and others. But I think he's really carrying the mantle in terms of ES or PSC differentiation toward primordial germ cell and other gonadal cells. And he showed some, he went through his whole, litany of many contributions with live pups everywhere. It's like making ES cells into live pups. That's just normal now, which is insane. But I showed also some unpublished work with live reporter embryonic stem cells for evaluating sex determination, and showing a lot of mechanism here along the way of making testicular organoids. And this is kind of him closing the loop, that now he can make all ESC ovarian organoids, testicular organoids, which includes both the primordial germ cell-like cells and their gamete derivatives, as well as the gonadal support cells that are necessary in order to induce them, where you used to have to get those from fetal tissue, which was just not practical, all in mouse, mind you, but he has also done some work in human. The data he showed is all in mouse, but he's closed the loop in mouse, and that's the key, with demonstration of live-born pups using material that's entirely derived from embryonic stem cells. So this is, I think, a long sought goal that we've reached, and now it's a matter of translating it to the human system in a viable and practical way. Although, as Arun will probably tell you, I don't think we should ever be making gametes from stem cells, but that's a whole other story. We can. All right, that's the thing. We can talk about whether we should another time, but it looks like we're moving towards the capability. And yeah, just finally, this mechanistic and I think conceptual insight that it's long been known that kind of in terms of the sexual discrimination, that femaleness is suppressed by just two genes on the Y chromosome. Here, he made this novel insight that it goes the other way as well, that maleness is suppressed by some oocyte-specific genes. So it's this kind of bi-directional repression that reminds me a lot of my marriage, Arun. What did you think about those late concurrents?
No comment on your marriage, but I thought they were great. You moved around, I stayed put, and the concurrent session focused on novel approaches to program and perturb cell fate. This is a really neat session, highlighting some new advances in direct reprogramming, the ways basically to turn cells from one cell type to another. Of course, highlighted overall by IPS reprogramming, which is the probably the most high profile way of direct reprogramming that we know about these days, right? First talk was from Felipe Pereira at Lund University, using combinatorial transcription factors to reprogram cell fate. And this is a concept that has come up on our show a couple times. Different forms of transcription factors targeted towards different cell types, seeing how you can accelerate perhaps the production process of these cells. This was a situation where they used reprogramming, direct reprogramming, as a way to develop new immunotherapies. So, very translatable here. First, they used a computational approach to try to figure out the right transcription factors associated with 71 different immune cell types. And let me tell you, this is why I'm not an immunologist, because 71 cell types scares the crap out of me. All right. The name for the system is repro code, which I like. And it's useful for, like I said, inducing different types of immune cells, including dendritic cells, at really high capacity. I really like this system. Moving on to Matthias Ilmari Altio. This was one of my favorite talks because seems like such a powerful tool. He was focusing on a paper that he had published recently in eLife. He's from the Genome Institute of Singapore, an A-star. The title of this paper is Genomic Safe Harbor Loci and Landing Pad Cassettes for Reprogramming iPS-derived cells. We all know about that safe harbor locus AAVS1 that is in iPSCs, and we can use it to introduce different genomic elements for overexpression and so on. But here, he he actually led off with a talk saying he doesn't like AVS1. It's not the safest of the safe harbor loci. So using computational approaches, again, a common theme here, he found 25 different unique safe harbor sites that haven't been reported before, and actually narrowed that down further to three that were validated using various criteria, using these landing pad cassettes, harboring a GFP reporter, and ultimately differentiating into iPSCs, doing the standard mesoderm, ectoderm, endoderm characterization. So I think this is a immediately translatable tool that perhaps everybody in the field could use because we all know the pros and cons of AAVS1. And it's nice to have multiple options in terms of safe harbor sites. And perhaps these three are the new options, right? Moving on to Stefan Yarabek from Columbia University. This was I don't want to say controversial, but had the potential to be controversial. But he'll be the first one to tell you about that. This is, you can see it in the title, Base Editing in Human Embryos of PCSK9. All right. We have talked about the potential and the ethical controversies associated with embryo editing, human embryo editing in particular. And of course, he was approaching this from a purely basic science perspective and gave a very nice disclaimer about the implications of his work at the end of the talk, saying this is not ready for prime time clinically at all. But here he was looking at whether base editors, which have been very powerful recently, could be a potentially cleaner way to edit early human embryos at the zygote level for research purposes, and was able to get 70% editing efficiency for the HGB1 and 2 loci, but still some indels, unfortunately, introduced because you're using a Cas9 system. Even though it's the adenine base editing system, it's built around the Cas endonuclease, which is somewhat of a dirty system, as we know. The PCSK9 locus was also edited and actually had a little higher specificity and efficiency in editing that locus. They were able to carry these forward to actually blastocyst level embryos and make actually edited human embryonic stem cells from these base editors. So, a really cool tech driven story. And I like how he framed the ethical part of the work as well at the end of the talk. And then I wrapped up with Camila Vasquez Echegaray also from Lund University, talking about avenues for interrogating cell plasticity barriers in complex biological processes. And again, this is a similar talk to the one I talked about before, where they were using a combinatorial transcription factors and combinatorial new factors for cell type specific reprogramming. And so that was the end of the concurrent sessions for that portion of the day. We actually shifted to another session that we love to do every single year, which is our annual meetup hub with the fans and listeners of the Stem Cell Podcast. Always great to chat with you guys. Thank you so much for listening. We do what we do thanks to you.
Yeah, I mean, between that and the delegate session was nice too because you had like one-on-one where you could talk to them at length. We had like 10 minutes with everyone. I get so anxious when there's like, I mean, not like there was a big crowd to come meet us, full disclaimer, but I just feel I would hate to have someone wait to talk to pitiful old me. But it was great. I mean, when we got the conversations going with the attendees to the meetup hub, it was just, once you get over the initial awkwardness, it's just so interesting and fascinating to talk. to the fans, I guess you would call them, from all walks of life, and really inspiring. Makes me want to keep doing this podcast. I mean, we came straight from there to here, and I am full of vim and vigor, and not least of which because we're about to get back downstairs and do this live show, which is going to be a real standout at this meeting, and I mean for the podcast history, to be honest. So really looking forward to that. Another element of the show that really gets us closer to the fans, which we love. So thanks guys for coming to that session. Thanks in advance for coming to the show tonight. And it was really great meeting everyone at the delegate session, if that's what we're calling it, yesterday. That show is going to come out in about a month. I can't wait for all this product to hit the airwaves. Hoping you guys will enjoy it too. But for now, that brings us to the end of our latest ISSCR 2024 episode. Don't forget to follow us on X at STEMCELL Podcast. Find out what we're up to at the meeting. You can also check back here tomorrow for our final recap, day 4 of the meeting, which we're gonna tape right before the awards session and send directly out to you guys. Can't wait for that. And really, so far, this has been a delightful meeting in a delightful city. And we hope that you guys are enjoying it too.